You already know that a living organism is a collection of organic molecules. All living organisms are composed of one or more cells, which are considered the smallest units of life. The compounds in a cell mostly consist of carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. So, isn't it amazing that a living organism is made of non-living atoms and molecules? The study of the cell at the molecular level falls in the domain of biochemistry, which is a very important branch of biology. It is equally an important branch of organic chemistry too. In general, biomolecules are bigger and more complicated. So, let's quickly reinforce our knowledge of basic organic chemistry by applying it to these complex substances. Let's first define the term biomolecule. A biomolecule is a chemical compound that naturally occurs in living organisms and which are necessary for the existence of all known forms of life. For example, keratin. The main component of hair is an agglomeration of proteins. Based on molecular mass, biomolecules can be classified into micromolecules and macromolecules. Micromolecules are biomolecules whose molecular weight is less than 1000 and include sugars, lipids, vitamins, and so on. On the other hand, macromolecules are biomolecules whose molecular mass is greater than 1000, which includes proteins, polysaccharides, and nucleic acids. Sugars and proteins are essential constituents of our food. These biomolecules interact with each other and constitute the molecular logic of life. Carbohydrates are a vital source of the three basic necessities of life, that is, food, clothing, and shelter. Carbohydrates constitute a very large group among various naturally occurring organic compounds. In nature, they are primarily produced by plants during photosynthesis. Some common examples of carbohydrates are cane sugar, glucose, and starch. Carbohydrates got their name because historically they were considered to be hydrates of carbon. Carbohydrates are also called saccharides, from the Greek word saccharon for sugar. Many, but not all carbohydrates have the general formula CxH2OY. For example, glucose C6H12O6. Its formula can be rearranged to fit the general formula. So, is CH3COOH considered to be a carbohydrate? Perhaps you recognize this compound as acetic acid. Its formula does fit the pattern, but acetic acid is a carboxylic acid, not a carbohydrate. The formula isn't enough to define a substance as a carbohydrate. To be more precise, we will define carbohydrates as optically active polyhydroxyaldehydes or ketones, or compounds that produce such units when hydrolyzed. For example, glucose is a polyhydroxyaldehyde, while fructose is a polyhydroxyketone. Carbohydrates can be classified based on their behavior during hydrolysis. These broad groups include monosaccharides, oligosaccharides, and polysaccharides. Monosaccharides are the basic building blocks of all carbohydrates. Monosaccharides cannot be further hydrolyzed to give a simpler unit. About 20 monosaccharides exist in nature. Glucose, fructose, arabinose, and ribose are some examples of monosaccharides. Oligosaccharides yield 2 to 10 monosaccharide units when hydrolyzed. On the basis of the number of monosaccharide units that are produced on hydrolysis, this group of carbohydrates can be further classified into disaccharides, trisaccharides, tetrasaccharides, and so on. On hydrolysis, a molecule of a disaccharide yields 2 molecules of monosaccharides. In other words, a disaccharide is made up of 2 monosaccharide units. The monosaccharide units that are produced on hydrolysis may be the same or different. Sucrose, lactose, and maltose are examples of disaccharides. On hydrolysis, a molecule of sucrose, commonly called cane sugar or beet sugar, produces a molecule each of glucose and fructose. But a molecule of maltose produces 2 molecules of glucose. Raffinose is a trisaccharide, while stachyose is an example of tetrasaccharide. Polysaccharides yield a large number of monosaccharide units when hydrolyzed. Polysaccharides, which include starch, cellulose, glycogen, and many gums, do not have a sweet taste. Hence, they are referred to as non-sugars. We often use the term sugar to refer to carbohydrates that have a sweet taste. In general, monosaccharides and oligosaccharides are sweet in taste and are therefore referred to as sugars. Everyday examples of sugars include sucrose, found in cane sugar, and lactose, found in milk. Which of these structures is a monosaccharide? Which is a disaccharide? Can you identify an oligosaccharide and a polysaccharide? Molecule A is a polysaccharide. Molecule B is a monosaccharide, while molecule C is an oligosaccharide. Molecule D is a disaccharide. Carbohydrates can also be classified as reducing sugars and non-reducing sugars. Reducing sugars are carbohydrates that reduce Felling's solution or Tollens reagent. On the other hand, non-reducing sugars are carbohydrates that reduce neither Felling's solution nor Tollens reagent. All monosaccharides are reducing sugars. Sucrose would give negative results with both Felling's and Tollens test. Hence, it is a non-reducing sugar. Most disaccharides are reducing sugars, and sucrose is unusual in this respect. Monosaccharides can be further classified into two major categories on the basis of the nature of the carbonyl group present, as aldoses and ketoses. The term aldose is used to describe a monosaccharide that has an aldehydic group. The term ketose is used to describe a monosaccharide that has a keto group. For example, glucose is an aldose, while fructose is a ketose. Aldoses and ketoses can be further classified on the basis of the number of carbon atoms present, as shown here. The prefixes tri, tetra, etc. with the ending -ose indicates the number of carbon atoms present. For example, glucose is a monosaccharide that has 6 carbon atoms with an aldehydic functional group. Hence, it is an aldohexose. Similarly, fructose is a monosaccharide that has 6 carbon atoms with a keto functional group. Hence, it is a ketohexose. Can you classify this monosaccharide based on the number of carbons and the functional group? First, by counting along the carbon skeleton, we see that there are 7 carbon atoms, a heptose. This molecule is one of the few heptoses found in nature. This sugar contains a ketone group, so it is a ketose. Combined, we can fully describe this as a ketoheptose. Glucose is widely found in nature in the free as well as in the combined state. In the combined state, glucose occurs in cane sugar, starch, and so on. It is present in sweet fruits as well as in honey. Ripe grapes are also rich in glucose. Glucose is the monomeric unit of the larger carbohydrates such as starch and cellulose. Glucose is an aldohexose, which is probably the most abundant organic compound found on the earth. Glucose is also called dextrose because aqueous solutions of glucose are dextrorotatory in nature. That is, an aqueous solution of glucose rotates plane polarized light towards right. Let's now see how glucose can be prepared in the laboratory and on a commercial scale. In the laboratory, glucose can be prepared by the hydrolysis of cane sugar. An alcoholic solution of cane sugar or sucrose, on boiling with dilute hydrochloric acid or sulfuric acid, produces equal amounts of glucose and fructose. On a commercial scale, glucose can be prepared by the hydrolysis of starch. Starch on boiling with dilute sulfuric acid at 393 Kelvin under a pressure of 2 to 3 atmospheres produces glucose. Let's now study in detail the determination of the open chain structure of glucose. Glucose is known to have the molecular formula C6H12O6, as shown by elemental analysis and molecular weight determination. The next step is to figure out its molecular structure and absolute stereochemistry, which were very challenging tasks. Let's consider some evidence that helps scientists to determine the structure of glucose. For example, the reaction of glucose with hydrogen iodide, glucose on prolonged heating with hydroiodic acid in the presence of red phosphorus, results in the formation of N-hexane. The formation of N-hexane indicates that all the 6 carbon atoms in glucose are linked in a straight chain. The reactions of glucose with hydroxylamine and hydrogen cyanide also provide similar evidence. Glucose reacts with hydroxylamine to form glucose oxide, which shows the presence of a carbonyl group. Additional evidence supporting the presence of a carbonyl group is provided by the reaction of glucose with hydrogen cyanide. Glucose reacts with hydrogen cyanide to give glucose cyanohydrin. When glucose reacts with bromine water, a mild oxidizing agent forms gluconic acid, which has the same number of carbon atoms as shown here. This is only possible if glucose contains a carbonyl group at the terminal position. That is, if the carbonyl group is an aldehydic group. Further, as the keto group doesn't respond to mild oxidizing agents, we can confirm that the carbonyl group is an aldehydic group. The acetylation reaction of glucose with acetic anhydride results in the formation of glucose pentaacetate. This indicates that 5 hydroxyl groups are present in a molecule. As the pentaacetyl derivative formed is stable, it means that the 5 hydroxyl groups are attached to different carbon atoms. The oxidation of glucose with nitric acid, a strong oxidizing agent, yields a dicarboxylic acid called saccharic acid. Gluconic acid also forms saccharic acid under these conditions. These reactions indicate the presence of a primary alcoholic group. Thus, based on these reactions, we can say that a glucose molecule has an aldehydic group and 5 hydroxyl groups, including 1 primary alcoholic group and secondary alcoholic groups. Hence, the open chain structure that can be assigned to glucose is as shown here. As this structure possesses 4 asymmetric carbons, there are 16 possible stereoisomers. Emil Fischer established the configuration of naturally occurring dextrorotatory glucose. It was truly an intellectual feat to identify which of the 16 possible stereoisomers of aldohexoses matched the structure of naturally occurring dextrorotatory glucose. Fischer won the Nobel Prize for this in chemistry in 1902. Fischer's projection formulae are widely used to represent the structures of carbohydrates. To assign the configuration of carbohydrate containing more than 1 chiral center, the lowest asymmetric carbon of a carbohydrate is compared to the configuration of an arbitrarily chosen standard, that is, dextrorotatory glyceraldehyde. Glyceraldehyde is chosen as a standard of reference as it is the simplest carbohydrate that has a single asymmetric carbon that shows optical isomerism. Shown here are the 2 enantiomers of glyceraldehydes. The carbohydrate or monosaccharide that can be chemically correlated to the plus isomer of glyceraldehyde is said to possess D configuration. And the carbohydrate or monosaccharide that can be chemically correlated to the minus isomer of glyceraldehyde is said to possess L configuration. That is, the compounds that are formed as a result of a reaction or a series of reactions from glyceraldehyde without breaking the bonds around the chiral center or any sort of other chemical correlation are assigned the same configuration as that of the particular optical isomer of glyceraldehyde. For example, the levorotatory lactic acid formed from dextrorotatory glyceraldehyde with D configuration is also said to have D configuration. It is important to note that D or L represent the relative configuration of a particular stereoisomer and doesn't hold any relation with the optical activity of the compound. To find the configuration of a monosaccharide relative to glyceraldehyde, compare the configuration of the lowest asymmetric carbon to the two glyceraldehyde structures as shown here. If the configuration of the lowest asymmetric carbon matches the spatial arrangement of the plus stereoisomer, that is, if the OH group is on the right-hand side, then the molecule is said to have D configuration. For example, the molecule shown here is D-glucose. This molecule is dextrorotatory glucose with D configuration. If the configuration around the lowest asymmetric carbon matches the arrangement of the minus stereoisomer of glyceraldehyde, that is, if the OH group is on the left-hand side, then the molecule is said to have L configuration. The molecule shown here, then, is L-glucose. This molecule is levorotatory glucose with L configuration. Here is a problem for you to try. Xylose and aldopentose makes up the polysaccharides present in corn cobs and the wood of trees, and is chemically correlated to dextrorotatory glyceraldehydes. The two Fischer projections of xylose are shown here. What would be the configuration of xylose? We know that the plus isomer of glyceraldehydes is assigned the configuration D arbitrarily. By comparing the configuration of the lowest asymmetric carbon in xylose with this glyceraldehyde, we can assign the configuration D to xylose. We have already discussed the open chain structure of glucose on the basis of several reactions. However, it was observed that the open chain structure couldn't explain certain properties of glucose. However, these limitations were overcome by the establishment of the cyclic structure of glucose that was proposed on the basis of several experiments and X-ray analysis. Let us first look at certain important reactions that were not explained by the open chain structure. The open chain structure of glucose has an aldehydic group. Hence, it is expected to give a positive Schiff's reagent test. However, it doesn't. Note that in Schiff's test, an orange-colored solution of fuchsin is prepared and then decolorized by reacting it with sodium bisulfite. If an aldehyde is added, such as acetaldehyde, then the solution will turn a vivid purple. Additionally, glucose does not form the hydrogen sulfide addition product when it reacts with sodium hydrogen sulfite. Also, the pentaacetate derivative of glucose does not react with hydroxylamine. This further suggests that there is no free CHO group in glucose. Another observation is that glucose is found to exist in two isomeric forms, alpha and beta. The alpha form has a melting point of 419 Kelvin. It is obtained by crystallizing glucose from a concentrated solution at 303 Kelvin. The beta form has a slightly higher melting point of 423 Kelvin. It is obtained by crystallizing glucose from a hot, saturated aqueous solution at 371 Kelvin. In order to explain these observations, which are not consistent with the open chain structure, a ring structure was proposed for glucose. The work done by many chemists helped establish the cyclic structure of glucose. It accounts for all the objections against the open chain structure. Since the ring structure has no free aldehydic group, glucose does not respond to certain characteristic tests of aldehydes, like Schiff's test and addition reaction with sodium bisulfite. On the other hand, typical aldehydic reactions, such as the formation of cyanohydrin and oxime, which involve the attack of HCN and NH2OH, is presumably due to the open chain structure of glucose that is formed by the opening of the ring. The ring opens when these strong reagents attack it. The cyclic structure is attributed to the formation of hemiacetal from the reaction between the aldehydic group and the C5-hydroxyl group present within the same molecule. The cyclic structure thus formed is a six-membered ring. This cyclic structure was experimentally elucidated by Haworth and further confirmed by X-ray analysis. This cyclic structure has one chiral center more than the open chain structure. Therefore, two possible isomeric forms are possible, as shown here. These two isomers differ in the configuration of the hydroxyl group at C1. Stereoisomers that differ in the configuration at C1 are known as anomers. C1 is called the anomeric carbon. Note that it is the aldehyde carbon before cyclization. The six-membered ring structures of glucose can be called pyranose structures, in analogy with pyrin. Pyrin is a six-membered heterocyclic compound. Hence, the anomers are called alpha-D plus glucopyranose and beta-D plus glucopyranose. Note that the hydroxyl groups on the right side and the left side in the Fischer projection formula are directed below and above the plane of the ring, respectively, in Haworth structures. Also, the terminal CH2OH is always projected above the plane of the ring. Let's direct our attention to the functional isomer of glucose, that is, fructose, another important monosaccharide. Like glucose, fructose has the molecular formula C6H12O6. Fructose is readily soluble in water. It is the sweetest of all sugars. It is obtained by the hydrolysis of sucrose with glucose. The chemical equation corresponding to this reaction is as shown. Certain characteristic reactions of fructose show that it is a ketone and consists of 6 carbon atoms in a straight chain with the keto functional group at position 2 of the carbon chain. Unlike glucose, fructose is observed to be levorotatory, that is, it rotates plane polarized light towards the left. In fact, an old name for fructose is levulose. Does the fructose structure illustrated here belong to the D or L series of sugars? As fructose can be chemically correlated to dextrorotatory glucose with D configuration, it also belongs to the D series. Hence, fructose is levorotatory with D configuration. Its open chain structure along with the name D minus fructose is illustrated here. As the open chain structure fails to explain certain facts like its existence in 2 isomeric forms and the formation of hydrogen sulfide addition product, the ring structure was established. The cyclic structure is obtained when the hydroxyl group at C5 gets added to the carbonyl group at C2. The cyclic structure thus obtained is a 5-membered ring. This structure has one chiral center more than the open chain structure. Therefore, there are 2 possible isomeric forms as shown here. The 2 cyclic forms differ in the configuration of the hydroxyl group at C2. These isomers are called anomers. The 5-membered ring structure of fructose is called furanose, in analogy with furan. Furan is a 5-membered heterocyclic compound. The furan molecule contains 4 carbon atoms and 1 oxygen atom. The cyclic structures of the 2 anomers are named alpha D minus fructofuranose and beta D minus fructofuranose. Disaccharides are carbohydrates that yield 2 monosaccharide units on hydrolysis. The hydrolysis of a disaccharide may yield 2 molecules of the same or different monosaccharide. In disaccharides, the 2 monosaccharide units combine and result in the formation of an oxide linkage with the elimination of a water molecule. For example, sucrose with 2 monosaccharides, glucose and fructose, joined together by an oxide linkage is shown in the structure here. Such a linkage, formed when the hydroxyl group of the hemiacetyl carbon of one monosaccharide condenses with the hydroxyl group of another monosaccharide, is called glycosidic linkage. In other words, a linkage between 2 monosaccharide units through an oxygen atom is called glycosidic linkage. The 3 most important disaccharides are sucrose, maltose, and lactose. Let us now focus our attention on their structural aspects. The well-known disaccharide plus sucrose or table sugar is obtained from sugarcane or sugar beets. It has the molecular formula C12H22O11. The hydrolysis of sucrose yields an equimolecular mixture of D plus glucose and D minus fructose. Solutions of sucrose are dextrorotatory, but when sucrose is hydrolyzed, it becomes levorotatory. There is an explanation for this anomaly. The optical rotation of D-glucose is found to be plus 52.7 degrees, and that of D-fructose is minus 92.4 degrees. That is, D-glucose is dextrorotatory with an optical rotation of 52.7 degrees, while D-fructose is levorotatory with an optical rotation of 92.4 degrees. As the magnitude of levorrotation of fructose is greater than the magnitude of dextrorotation of glucose, the mixture obtained by the hydrolysis of sucrose is levorotatory. The sign of optical rotation changes from dextrorotatory to levorotatory during the process of hydrolysis. This is called the inversion of sugar. The levorotatory mixture, which is a mixture of equimolar glucose and fructose obtained from the hydrolysis of dextrorotatory sucrose, is called invert sugar. Invert sugar is the main ingredient of honey. Let's look at the structure of sucrose in greater detail. Sucrose is made up of one unit of glucose, specifically the alpha-anomer alpha-D-glucopyranose, and one unit of fructose, specifically the beta-anomer beta-D-fructofuranose. These two hexose units are joined by a glycosidic linkage that is formed between the C1 of alpha-D-glucopyranose and the C2 of beta-D-fructofuranose. Note that the loss of a water molecule results in the formation of 1,2 glycosidic linkage or alpha-beta glycosidic linkage. Thus, the two carbonyl functional groups of glucose and fructose are blocked with the formation of glycosidic linkage. As the reducing group is necessary to give Tollens and Fellings tests, is involved in the formation of the glycosidic linkage, sucrose gives a negative test with these reagents. Hence, sucrose is called a non-reducing sugar. Maltose is another important disaccharide which contains 2 glucose units. Its molecular formula is C12H22O11. It is the least common naturally occurring disaccharide. It is found in germinating grain. The glycosidic linkage in maltose is formed between the carbonyl group of the C1 of alpha-D-glucose and the hydroxyl group of the C4 of the second alpha-D-glucose. The availability of a free aldehydic group at the C1 of the second glucose in its solution allows the maltose to reduce Tollens and Fellings reagents. Hence, maltose, unlike sucrose, is a reducing sugar. Lactose is a disaccharide present in milk, so it is sometimes referred to as milk sugar. Lactose is composed of beta-D-galactose and beta-D-glucose. The glycosidic linkage in lactose forms between the C1 of galactose and the C4 of glucose. Like maltose, lactose is a reducing sugar due to the presence of a free aldehydic group at the C1 of the glucose unit. Can you answer this question on your own? What are the products expected when lactose is hydrolyzed? Lactose would produce galactose and glucose in equal proportions. Here, we see the Haworth projections of lactose and the hydrolysis products. Polysaccharides contain many monosaccharide units joined together by glycosidic linkages. Polysaccharides are the most common naturally occurring carbohydrates. A polysaccharide has the general formula C6H10O5 taken n times. They play important roles in food storage and as structural material. Cellulose and starch are the most important polysaccharides. Starch is the main storage polysaccharide of plants. Starch is found in cereals, roots, tubers, and some vegetables, and is used as an important dietary source for human beings. Chemically, starch is a polymer of the alpha isomer of D plus glucose. It consists of two components: amylose, which makes up 20% of starch, and amylopectin, which makes up the remaining 80%. These two components differ in their molecular size and shape. Amylose is the water-soluble portion of starch. Amylose molecules contain 200 to 1000 alpha D plus glucose units, connected by C1, C4 glycosidic bonds, as shown in the illustration. The chains of amylose are long and unbranched. Amylopectin, the major component of starch, is insoluble in water. Amylopectin is a branched chain polymer of D-glucose units. The branched structure consists of several hundred short chains of about 20 to 25 D-glucose units each. An amylopectin molecule may contain as many as 1 million glucose units. The main chain of amylopectin contains alpha-1, 4 linkages between the glucose units, while branching occurs by alpha-1, 6 linkages, that is, C1 to C6 glycosidic linkages. Consider the illustrations here. Which one best represents amylose? Which one best represents amylopectin? You just learned that amylose is an unbranched chain, whereas amylopectin is branched. Therefore, picture A shows amylopectin with its branched chains. Picture B shows amylose. Note that the unbranched chains of amylose often adopt helical structures. Cellulose is another important polysaccharide. Found exclusively in plants, it is the most abundant of all carbohydrates. Cellulose is the chief structural material in plants. It is completely insoluble in water. Cellulose is a straight-chain polysaccharide. It is made up of chains of beta-D-glucose units. These beta-D-glucose units are joined by glycosidic linkages between the C1 of one glucose unit and the C4 of the next. These are also called beta-1,4 linkages. Humans cannot digest cellulose, as they don't have the enzymes necessary to do so. Cows, horses, goats have symbiotic bacteria in their intestinal tracts. These bacteria provide the enzymes for breaking down cellulose. The term dietary fiber refers to cellulose. The polysaccharide, glycogen, is the form in which carbohydrates are stored in animals. It is present in the liver, muscles, and brain. Glycogen is also found in yeast and fungi. The structural aspects of glycogen are much similar to amylopectin, a component of starch. Hence, it is referred to as animal starch. The structure of glycogen differs from that of amylopectin in that the molecules of glycogen have a greater degree of branching. Enzymes break down glycogen that is stored in the liver and muscles into glucose when the body needs it. Now let us study briefly the importance of carbohydrates in our lives. Carbohydrates are a very important class of biomolecules. They form a major component of our diet. They function as storage molecules, starch in plants, and glycogen in animals. The cell walls of bacteria and plants are made of cellulose. Carbohydrates have many uses as structural material. We build houses and furniture and other useful items from wood, which is 40 to 50% cellulose. We clothe ourselves in cotton and linen, which are largely cellulose. Carbohydrates provide the basis for a variety of industries, including textiles, paper, lacquers, and breweries. Carbohydrates have a number of important biological roles. The sugars D-ribose and 2-deoxy-D-ribose are components of nucleic acids.